Okay, so today we are going to go through dimensionality reduction computer session and to use some real numbers to let you get the feeling of what's going on over there, how this projection of vectors into another vectors and help you to rotate and do this PCA. If you are in the Teams channel, you will find this outline for today's session. We will revisit the projection of one vector to another, and then we will see how to compute the covariance matrix, and then through that to use the covariance matrix to compute the PCs. And then we play with this built-in function in MATLAB and try different outputs variables and see what they mean. How can you use this output for your next step? We will have a t-SNE function and also run UMAP. And through the interface of the single cell gene expression tool, we can do all this using real data, okay, including fate. So the first, let's how many people are you here? By the way, the second homework I haven't released yet. I'm going to give you the online assignment instructions for the second assignment later today. Basically, it's take a video and explain what the PCA is and how it works. Okay. So as I said, I showed this many times already, and we just one more time using real data to see, okay, how can you compute the projection of vector A to the B? So the idea is to use the B as a new coordinate system for A. Let's see this A it has a position. So this is the x-axis, our original. And this example of A will be this place. Say our example have A say 2 and 3. So we describe this position as a 2 and 3. That's because it's a projection on x-axis. This length is a 2, right? And this projection on this vector length is a 3. So that's why the position is described as a 2 and a 3. So on computer, if we define A, this small a, as a vector, so you have 2 and 3, right? So that's the position. Our rotation matrix by default is 1, 1. Okay, this is our original rotation matrix. So this is the first column here in my rotation matrix. Is this is my x-axis, okay? So 1, 0 gave you the direction, which is here, and the 0, 1 is the direction goes up here. So my rotation matrix is given by 0, 1. So if you use this rotation matrix to rotate this, the expression will be R times A, multiplied by A. Okay, R is a 2 by 2, and A is a 2 by 1. So the output will be 2 by 1, so still. So what you can see from here is I use this rotation matrix defined by these 2 vectors, blue one, applied to my vector. The output, the product of this is still its origin, and then nothing changed. The point was A 2, 3 is still 2, 3, because this is your original rotation. There's no change, it's still the same. What we want to make changes to define a different set of rotation matrix. So my green rotation matrix will be something like this. It requires and to be go this way, for example, and then the other one should be orthogonal to each other. So I would see, okay, this green one is my new coordinate system, and it's required to be something like a cosine theta, sine theta, x negative sine theta, cosine theta. This expression have has a meaning, because if I do this, it's a theta with a rotation from there. But you can actually give any pairs of data as long as you normalize it. For example, I'm going to give, this is a longer version, 5, 2. So my B well, this B is too long. It's not normalized. So I need to normalize it back. So it's have a magnitude to be 1. So how can you normalize B? So if I decide to use B as my new x-axis, B has to be normalized by the length. So B 2, and normalize it. So I need to replace this to be a shorter version, which 0.9285, 0.34. Okay, so you have to move this back to here. Okay. And you can compute some kind of theta related. So you can replace this with 0.9. Okay, so if I use this as my new axis, I need to compute, if I project my original point using this as my new axis, I need to compute the length of this. So this will be my new XAI. I need to know how long this is. So how to compute this? You just do A times B. Because according to this, the length of this part is A times B. This is how long this is. Okay. If you use normalized version, so A just times B normalized version. A times this, so this is a normalized version. if you times unnormalized version, the original B, so which you have A project on this is too long, then you just divide by the norm of B. You still get the same value. So how can I compute in our example how long this part is? So I will do dot product between A times normalized B, and I get the result. Or I can just A times unnormalized B, but divide by the B's norm. So how long this is, projection, I will do A times B, dot product. Oh, wrong. So I need to transpose this, so the length is 2.9. Okay, how long this is? 2.9. So how can I compute the projection of A to this vector? So you have, that's your new x-axis, this is new y-axis, new y, new x. You also compute the projection of A to here. So it depends on, you need this distance. So you need to have a description of this vector. So this vector will be negative 0.4. Okay. So once you determine this, the last one will be automatically defined. So you don't have to describe this as long as you found new x-axis and this y is orthogonal to each other. Automatically should be this. So depends on where. So what you can do now is to project A onto the new B x and new B y. So you compute both. Distance is D1, D2. So after rotation, this A coordinates becomes B1, D1, D2, distance D2. It was 2 and 3. Based on this coordinates, it should be D1, D2. Okay? So my new rotation matrix will be, so I have a B, so my new rotation matrix will be B, wrong one. Yeah, so I'm constructing here is this. So where this come from? That's from the random. I randomly picked this as a new axis. It was 2.5 normalized into 0.9, this one. And where this come from? This is just orthogonal to this x-axis. I have a y-axis automatically defined. So this is my new rotation matrix. Now I use new rotation matrix multiplied by with 2 my A. So now remember, if I multiply it by this, and I will still get 2, 1, 2, 3. But now if I multiply it by this new rotation matrix defined by this new system, and my coordinates becomes 0.7, something wrong. That's new position. Is that right? So, yes, according to this, I have the position for this new description is 0.7, 0.5. So A project onto the B. Let's see. Rotation matrix. There's something wrong with here. Let me do I believe this is the correct formula to rotate. Let me think how it's, because after rotation, this coordinate system should be 2.9 and 2.0. So this is the correct expression. I don't know how this rotation matrix. So this rotation matrix is, and I project A onto first, the second. So I need really to change this in order to get it correct. So somehow this rotation matrix needs to transpose in order to have this two multiplied by this number, because this number is vertical, and I need to rotate this. I need to rotate this R, so I can do that. Okay, I hope you get the idea. So to do the dot product for this with A, I need to rotate this. So I need to rotate this to the transpose in order to get a dot product between A and this one here. And then A with that one. So that's the correct formula to get the coordinates. So now we only rotate one data point here. But if I have several, say if I have this is A1, if I also have A2, let's put here A2. A2 is somewhere in here. Okay, if I project A2 into the new coordinate system, I will compute how long this is and how long this is. So let's make a number. So if I have this -3 and 3, okay? We use this as a new number. So if I have A2 is -3 +3, okay, at position. If I rotate A2 with this rotation matrix, it becomes -1.338. Okay, so this is a after rotation become that, depends on this coordinate system. So we have this A1 becomes, what's the A1? After rotation. So this becomes 2.97, 2.0. And this position was -3.3. Based on the new coordinate system, becomes -1.6 and 3.8. So that's the impact of this rotation matrix to these two data points. Now we can put two data points together. So I have my A, I have my A2. So this forms two points. So that's my two data points. The first one, 2, 3. So here, another one, -3, 3. I'm going to do the rotation for both of them at the same time. So the formula will be R rotation matrix times A together. So this single operation, I rotate both points at the same time. So the small rotation matrix rotate 2 points at the same time to get to the results as they did independently, separately. But we don't have to do it separately. We can put all the vectors together, make a matrix, and they make it longer, and they, then one rotation matrix operation and change all the coordinates. Okay, now let's jump to this particular example. Now my data become not only A1, A2. Now I have 3 data points. Okay, this data have 3 columns. So I'm going to rotate using E. Okay, I will project 3 points onto the E and to see how long the projection of each point onto the E itself. So here is the data, and here normalize the data to make it centralized, and then you have an E over there. Okay. So what I will do, I will type this data in 3 points. Okay, I have 3 points in the space. So I'm going to centralize them to make them move to the center. Data minus mean data. Okay. So that's what we get. Oh no. We need to get 2. Okay. So this means if you don't give this 2, it will compute vertically. That give you 3 data points. X, Y is average, which not what we want. We need to have this x-axis average, so data minus that, and this x-axis, y-axis. This is the correct way to centralize them. So the centralization doesn't do anything to the data. It's just a move. The data was somewhere there, but now we move to data 1 is here, another here, and then another here. So this is the V1, V2, V3. Okay. So the center of this 0, 0 was somewhere else, and now we just move back to the center to so the first column is V1 and second column V2 and V3. Now we are going to rotate it. We rotate, let them to project onto E, which is defined by these 2 numbers. Okay. So the 0.2, and so somewhere to go to this direction. That's my E. So I can also define by another. So there's a E1 and I have a E2, which also not to each other. But for simplicity, we only see. Okay, only look at this. What we want to compute now is to project each point onto E. For example, project this to E, project this to E, project this to E. Then compute how long the projection is. We will compute how long this part is, how long this is, how long this distance. D3. This is D1. This one is close to it, so there's a D2. Just think about like a shadow, okay, onto it. So how we compute that? We just do the using dot product. So we can have a V project onto the E, okay? The dot product so the same idea. So this is after your rotation, you use this as an x-axis, and the new coordinate system for V3 becomes 0.4. And for this becomes 0.9, and that's for so there's 3 numbers are there how long the projection. Now we get a square root for a squared number and then also sum them together. So we basically add up the distance, the three distance altogether to represent the dispersion level. So then we divide by 3. So this is let me see. Yeah, we need a dot here. So this number is a D1, D2, D3 squared and put together. We have 0.4138. And it's average because you divide by 3. If you have many points, then this distance will increase. So you take average. So this is the average diversity of all the data points once they project onto the E and how separate they are. So we want to make this bigger. We want to rotate the E in order to make this as big as possible. So maximize this total. We want to have bigger value here, and if you rotate, if you choose different E, and you have different sum of total. Now, we are using a different way to compute this number. This time, we are using covariance matrix. So to compute the covariance matrix, we just do something like this. This is our covariance matrix. What I'm showing you on the board, and also we had just previously have done this to use a projection of the length to compute this number, the diversity. Now I use a completely different approach, which compute the covariance matrix between variables. You have x, y, so two variables. So your covariance matrix looks like this, and you can confirm with the built-in function, the covariance matrix V. If you do this, you get exactly same covariance matrix. So just confirm you can compute covariance matrix using this formula. So where this formula come from, if you have to refer back to how covariance matrix is compute. It's somewhere here, okay? It's covariance matrix, how to compute. You see this expression. So if you have a normalized, centralized data, and then you do this. Transpose the data and then multiply it by itself, you get a covariance matrix. So that's what we are doing here. So transpose your data, centralize the data, then times itself, and you got a covariance matrix. And this covariance matrix will help us to compute this number in a different way. Okay, so if we have this, we can compute C times E. You got the same number because this interrelationship between covariance matrix and those projection results. Okay. Numerically, they are the same thing. And this, look at that, and you compute the dot product, you get the same number. So we want to maximize this number. So we want to change the values in the E, which is the rotation, the angle of this, and in order to get the largest value here. So if we can change the E, get the largest value here, that means we get the largest projection of the data onto the E. Yeah, I know there's a lot on this slide, but that's a way to try to help you to link why this covariance matrix of the data can be used to compute how to separate data points once they project on the new coordinate system. Why you can link the covariance matrix with this separation, the diversity, total diversity. Okay, so now let's move to according to our agenda. Oh, by the way, I do have two YouTube links to help you. For example, the first one to understand the covariance matrix and the PCA. You may want to spend some time there. You still get confused. What's going on? For the rotation matrix, there's a very good lecture when this professor from the Washington University, and he explained already 9 years ago, so he's getting older as well. So he explained this rotation matrix in the context of introducing singular vector decomposition. But he described this very well, and you should at least spend some time to watch these two videos to help you understand the covariance matrix, PCA, projection of those concepts better. Now we move to just look at the built-in function in the PCA function and how MATLAB implements all this. Okay, so if you do doc PCA, let's see where PCA function is located. You know how to build a function. Last week we have my script, my function to generate random numbers, but you can have other people develop the function. For example, PCA is located in a toolbox, in the statistics toolbox, as a function called PCA.m. So this is no difference from what you created last week, my function.m. So they are just different functions. What we can do is to look at the PCA documentation, and we are going to use examples in there to show. Okay, so PCA will be give you 3 data points, 3 or more. You need to find a rotation matrix, which is defined by the E1 and the E2. So for the PCA, your input will be your X. The PCA, your input will be the data points. Okay, could be V, or we use big X. Your output is a rotation matrix, the R. So you have rotation matrix, in our case, defined by the E1, E2. So this R is E1, E2, 2 vector, the 2x2 rotation matrix. Then you will use rotation matrix to rotate your X, depends on how to write it. It could be something like this. So you apply rotation matrix to your data to change and to get a rotated version of your data. That's your y. Your x becomes y, and nothing changed, only the perspective. It was, in our first example, it was a 2, 3, and it becomes 0.9 something. You change the coordinate values, but the data point is still the same. Another is still the same. The only changed its perspective based on your newly selected rotation matrix, the coordinate system. So x will be rotated into a y. You still have the same number of the rows, same number of the columns. Your input matrix doesn't change, and you only get the same input as output, but change the perspective. So your input is, for this, is your input is x. You will get output, the coefficient, as a rotation matrix. So that's our, and then you can have another, which is score. Score is your y. It's your, after your rotation, and what your x becomes too, okay? Let's use examples to illustrate this. We can move down and then use MATLAB provide example. So it will load. So let me clean this. This is a data set name, so their file called MAT. So this is a data file. So we load this in, and then we get some variables. Okay, this is our input. So we are going to use this ingredients. So you can have a look of these ingredients there. The data is looks like this. There are 4 numbers, so this is a 4-dimensional data ingredients. The small data set with the 13 data points. So our example is 3 data points and in 2D. And this example is 13 data points in 4D, okay? Because you have 4 columns. So as I said, if we consider this as x, that's our input. So I just rename it. So our output of PCA x. So what our output should be? The PCA output should be a rotation matrix. So can you tell me what's the dimension of R? In our case, this is a 2D, so our rotation matrix is 2 vectors defined by 2 numbers. So our rotation matrix is 2 by 2. So what's the output for this? This rotation matrix will be 4 by 4 because you have 4D data. So you have 1, first, second, third, and fourth dimension. The rotation matrix to have the power to rotate the 13 data points. So we can see, okay, this is a 4 by 4 matrix of a rotation matrix, right? It was computed through this covariance matrix eigenvalue decomposition. Then you can get this. Let's see how the example they use. So this is 4 by 4. It's just a different name. In our case, R is rotation matrix, and this is a coefficient. Don't worry about those. Yep. Okay, so the documentation gave a lot of special usage cases. We don't have to follow them. But here is the idea of what's this dimensionality reduction. So if you want to visualize this original X, because it's 4D, so we cannot visualize them all at the same time, but we can do like scatter plot, 3D. We can see X, the first column, X, the second column, X, the third column. Yeah, let's see whether I can use dot. The size. There are some options. Color, so I use red, too small. So I'm changing the size of those, say 15, and red color, and with the field points. So just want to make this more visible from the distance. So let's do 25. Still too small, the dots. To 50. So we have this 13 data points described by 4 numbers each, but we can only visualize 3D data, so I pick the first 3 variables. I can pick 1, 2, 3, I can do 1, 3, 4, or 2, 4, 2, 4, 3, so I have a combinations. It's kind of a feature selection. I can select the variable I need to visualize this data, okay? But no matter which one I choose, for example, here I choose 1, 2, 3, I can pick, say, 4, 2, 3. I use the last column as my x-axis value. I never be able to fully capture the potential variability in this data. I need to do PCA to reduce, to help me find the largest diversity among the data. So that's why I found this rotation matrix. I'm going to rotate my data into, so the PCA helped me find the rotation matrix R. I will use rotation matrix to rotate X, okay, to get out my Y, okay. I hope this works. I have to make sure the matrix multiplication can work because R is 4 by 4, and this 4 by 4, I need to transpose this because X is 13 by 4. It only work. So make sure I need to transpose this into 4 by 13. My result will be 4 by 13, and then I will transpose it back because I want Y have the same dimension as X. So after this, I get the rotated, okay, rotated Y. But the Y, all the data points are all the same, but it's just rotate. Now from this perspective, if I look at 1, 2, 3, the first 3 columns, then I should have this largest separation because the angle I pick for the Y after rotation, the first should be the largest. A new figure. So this is the projected. Now I can safely pick 1, 2, 3, the top 3 as my visualization. So because they are carefully picked by the PCA. These top 3 PCs will help you to see the separation of data. You can do the 2D if you do only scatter without the third column. So that will be the first PC and second PC to separate the data. Okay, so if you don't want to compute Y by yourself, you should be able to use the second output, so score. And this score should be our Y. Okay? There's some of this direction difference, but they are the same. So now you can use a score. Oh, very different. Let's see something. Oh, I need to do the centralized. So, so my Y hasn't been centralized. So there's some problem. So this may be not the exactly solution, but the idea is there. So to use rotation matrix to rotate X, but the same X need to be normalized somehow. But you can use the score as your output, as your rotated results. Okay. If you do 3D, you should have this kind of separation. Yeah, that's your data, 3D, after rotation. Okay, let's have 5 minutes break, then we come back to see how t-SNE works. Okay. Okay, so the PCA is to give you a rotation matrix, so you can apply this rotation to X. So this projection will be used as Y, okay? So this Y, then you start to visualize Y1 and 2 to first two dimension, and then to look at the distribution of your data. Well, you cannot do this for randomly pick any two dimension from the X itself, but after the rotation, this first and second, or if you use 3D, you can have a 3 columns information from the Y. And remember, this X and the Y has the same dimensionality. So if you have 1,000 data points with 1,000 by 5 columns, and your Y is still 1,000 by 5 columns. So for our single cell data, so if you do this, remember this is, say, 10,000 by 10,000. So our the input, and this rotation matrix will be 10,000 by 10,000 because the rotation matrix is equal to the number of this, how many variables you describe your. So if you, this is considered as genes, cells. Our toolbox, not using cells by genes, we have a matrix which is always the genes by cells. So that's our input by default. So if you gave this directly to the PCA function, you will mess up with the variables and the samples. So we need to transpose this to be a cell by gene in order to put into here, okay? Just make sure you understand which is the sample, which is the variables to describe each sample's property. If you make it wrong, then your rotation matrix will be in the wrong dimension, and then everything changed. So if you put this upside down, or not upside down, rotated, transposed, then you will get a PCA not for cells, but it's for genes. It still makes sense sometimes. You want to see, okay, these two genes always similar to each other. Depends on your question, okay? Now we move to the t-SNE. t-SNE, if you have a search, T-S-N-E, and it's another function in M file. So t-SNE, what we can do to do this documentation read, very easy, and have X in and Y out. Well, this Y will not be the same as your input. It will be a smaller number, depends on how many dimensions you want to keep. So t-SNE will learn the relationship between your cells, between your samples, then try to create a new low-dimensional space and put your samples in this low-dimensional space to keep their relationship. Statistically, the probability of the neighborhood will be kept. That's why this will be a reduced dimension space, and you will still have the same number of samples. Your data points numbers will be the same. So remember also this input will be sample by variable. So if you have gene by cell matrix, you need to transpose before sending into here. So we can use examples. If you copy the fishery data and their if you copy this to load your example data here, close all, clean all. Okay. So your facial data contains 150 samples with 4 numbers. That several different sets of the fish species. And then you use t-SNE to do this. Remember, your input is this measurement. The measurements is given by fish sample and the variable measurements. So you can directly put that in and you will get y. And y will be 150 rows, because you have 150 species samples. Now you can very quickly to visualize this by using grouped scatter plot. And the species, the name has been predefined because the people perform the measurement. They know each sample, 150, each of them, what species they are. So they gave 3 different names of the species, and you can see the result of the t-SNE of this XY coordinates for each sample. They automatically group the same species because they have similar measurements in terms of 4-dimensional data. They are similar to each other. This Setosa fish was grouped in this corner. And another 2 species, they are not clearly separable, but still form their isolated, somehow the isolated groups. So that means the t-SNE and using the measurements to group fish in a data-driving approach, because the t-SNE have no information about which species each sample is, but the results fit our pre-labeled sample very accurately. Basically, it's very separable. You don't see any yellow-orange fish have been grouped in this place because they are in this 4D space. They are not neighbors to each other. We can do PCA on this as well. So let's try PCA. So if you have Y, so PCA, the first we need a rotation matrix. Maybe we just score directly compute PCA and our measures. Okay. So we don't need the rotation matrix because this, we don't have to rotate ourselves. The score will do the job. Now, if we do the scatter plot using the scores of PCA, that's a PCA solution. You can also separate the blue fish from this 2 orange and yellow fish, and they're separable definitely in their space. So PCA also do a good job here, okay? But not as clean as this tightly grouped t-SNE separation. If your purpose is try to group cells by types, so t-SNE will have a better resolution. Any questions here? We don't have a UMAP built-in function in MATLAB. So for the UMAP, you have to use the toolbox I installed. So UMAP. So that's our UMAP. So Y2. So we will produce. Remember to do the transpose, because this function I make is based on gene to cell. So that means my input should be variable in the rows, samples in the columns. So this function requires the input have a opposite dimension required by t-SNE and PCA. So that's why I need to transpose this. Let's try this to see whether UMAP can run on this sample. And then I get the coordinates from the UMAP, and then we can do a G-scatter. So this time I use coordinates generated by the UMAP, which is Y2. Okay. So you see all these functions, PCA, t-SNE, and UMAP, they all take this raw data input matrix. You just make sure it's correct. You know whether you should put the samples in the rows or in the columns. For this, you need to transpose your input. The output, if you write it this way, it gives you the position or coordinates for the first. So the t-SNE will give you the projection of your raw data into the new space. And this one will give you, by default, the 3D projection, where t-SNE give you 2D. You can ask for 3D. Anyway, so once you have coordinates, you can start to use them to plot the position of your samples. Okay, so if you don't have a name of species, because you don't have a group IDs for each fish, then you cannot use G. You just use scatter without the G. G is a grouped scatter plot to show you different colors. But if you don't have any information about your species, you only use scatter and to plot their positions, and then help you to decide which group is which cluster, and you can assign them later on. So that's what I will try to see. If you do scatter, then you have this. Okay, the position of each fish is the same as this one, just no color, because there's no information about the labels of each fish. All right, if you have no question, we are going to move to the interface and use real data to see how this dimensionality apply to single cell data. So I will load example data, and in this case, I'm loading unprocessed. So I haven't done any dimensionality reduction, so I don't know the position. I just randomly put the cells in this space. Okay. Now we can try first is to export this data into the workspace. Our data contains X, which is a matrix, and a gene. So I want to have both available so I can work on them in the workspace. So now I'm export two variables. I take this with the gene name. Gene name may be not necessary, but X is what we need. We want to use it in our command line window. So if you click this, you should have this variable X here. Okay. X is a sparse matrix, so we need a function to restore it to full, because it's in its original form. I remove all the zeros because there's a lot of zeros in a compressed version. But then now, because we are going to use t-SNE and UMAP, those functions require this full matrix representation. So I add this function to convert the sparse version into the full version. Nothing changed. The information all the same. Yeah. We export from the interface. So if you still have no idea where the X is, because we load this example data. This example data contains big X. You need to do a save, then you export the data to workspace. Then you tick this. You tick this, and your X will be exported and save into a variable called X. Okay, to do PCA, we just run score. Remember, X need to be transposed because this is $7,000 genes over and 2,000 cells. Is by gene by cell, so we need to transpose it into cell by gene. So because that's required for the PCA, your samples has to be in the rows. So this will create this R matrix. See, R matrix is this, okay? If you look at the small dimensions to 2,000, that's not give you the full 7,000 by 7,000. It's truncated into this lowers the number into the samples. So this is because this is over dimensioned. So the dimensionality, the variable number is larger than your sample. So basically, it's a very long, very short sample, not enough, and dimensionality number is higher than samples. That's why you get this truncated rotation matrix. But anyway, the score will be it's 2,000. Let's see. Yeah, so the score is your 2,000 cells. Now we can have this figure, okay? You remember you can do you know how to do this scatter plot. Then so this our PCA solution for the first two PCs for your data. We just load into from a single cell. So each point is a cell. So this is not very helpful. We don't see structure. We see some structure from this perspective. We don't know how they are corresponding to different cell types. So we change our strategy to do the t-SNE. So why? The t-SNE, our input is X transposed. Okay. We should do a normalization transformation, but let's try what we can get. So this is a t-SNE solution by Y1, Y2. Okay, so side by side, this is our PCA solution, this is our t-SNE solution. So definitely this t-SNE start to show this potential clusters in the data. You also know how to run UMAP. So UMAP, you don't have to transpose your data because UMAP, this function, takes directly gene by cell matrix. So you don't see this apostrophe here. So we just run the UMAP, take some time. Oh, so the UMAP function killed my process. So I have to reload my data and come back to try UMAP. Okay, figure is my UMAP solution. Okay, this is my UMAP output. Figure, that's my t-SNE output. Okay, I don't know if you have this already, and let's try fade. Okay. This also have to be, you don't transpose this. Okay, if you want to follow the command line, so those are the ones I used. So this is the command line. I run t-SNE and show scatter plot. I run UMAP, scatter plot. Now it's a fade. So I have a Y, Y2, Y3. There are projections of the dimensionality reduction technique to the same data set. Just remember the input dimension has to be correct. Okay, this need a transpose. The others do not. Figure then this will be a Ty3, and this is the fate projection. Okay, I don't know if you get this, but you can come back to the interface, so you don't have to type those command line. Now you can have this data in there, and then we decide to do the embeddings. Okay, so we can have click this buttons. If you want to do them all, you can see with the Control Shift, with the Shift, then set them all. So with this, the program will run all of this T3 UMAP fate all together, and you can select the 2000 highly variable genes. So the program will do all these selected embeddings, 2D, 3D, all of them. The underlying function is what we had in the command line here. I just wrapped it up into this interface, so you don't have to type them. So all the embedding is done. So we have dimensionality on the raw data, and we want to show all these available results. They all same number of cells, different ways to do the embedding. 2D, 3D, once you have 3D, you can do the rotation. 2Ds for publication purpose. So you can compare, okay? This is the best way you can have your own data first. You want to know, okay, what's in there, what the structure looks like, whether there's one dominant group of cells and others noise. Or just like this, they're separatable subclusters in there. So fate is also suggests maybe there are some development trajectories in the data for cells from here become that. With this, you can, for example, you can brush cells. For example, I want to brush this group of cells. Use this, and I can brush. Okay, this group of cells has been projected by different technology in different place. So fate put this group of cells to here. You highlight it. You can rotate to see whether they're separated. They're not separated. Seems this is the end of a trajectory of this development. So we may be interested in what the other cells in here, this branch, this arm. So we can highlight those arms. And this gave you the idea of those cells, if you use different technologies, have been projected on a separate cluster. So it's this cluster in the UMAP and this cluster. Although they are far away from each other in the UMAP, but the fate suggests they may on the same development trajectory. Because if you look at this data, this is here, and this is here. So these two very far away from each other, but they may linked developmentally. Let's see what this is. So this is a major part. Okay, so this is a very powerful way. It's a multi-dimensional view at the same time to help you deep understanding your data points. On the market, I believe this is the most powerful visualization approach. I don't see any other languages can do this. Okay, you agree with me? Okay. So if you can make this and have some discovery in your own data set, it's a major contribution to the field, because this is a data-driven approach. Definitely, you see the relationship between seemingly unrelated cell populations. You may identify very rare cell type. For example, nobody have ever labeled this cell type in a biological systems. So maybe a new discovery. Who knows? It's all on here. And you know the relationship between these small clusters in the middle of the UMAP. Well, it's a different position in the fate projection. And you can see, okay, this is located in the center of this and that. So maybe it's a early stage of development in the tipping point of the development stage. So there are a lot of stories you can make through this visualization. On this interface, you have, you have all the projections already. So next time you don't have to redo the embedding. You just select. So I want to use UMAP 3D. So you select this, and your data will be the UMAP 3D projection. Then if you select, I want to use TSNE 2D, so that's your TSNE 2D projection, okay? There is a place where you can have a clean plot. So this is for publication purpose. If you select plots and then prepare TSNE, so this one you can copy and or save into. So for example, let's copy it. So I copy this and I can paste into PowerPoint. Yeah, so this one you can in your presentation or in publication. So that's the purpose of this preparation of the clean TSNE UMAP plot. Anything else? So now if you want to go back to the views, this is the big icon here. So you can click, say I want to have a multi-embedding view. So that will bring that back to the page. You just see here. Okay. And for this figure, you should have something like this. So you should be able to, I want to highlight one of two genes expression. You can select the gene from here. So I want to look at the ABCC, its expression. Yeah, so I need to find a way to do this color map consistent. And the same as the last one, have a color map different from others. So I will fix this problem in next version. So this, you can see the expression level for that particular gene. I see like this called ABCCB. It's highly expressed in some of these clusters, but not others. Okay. If you look at it, it's highly expressed in this group. So you can use this to start to look at the genes and find subclusters have a high expression of this particular gene. So once you do this many times, try different genes. Most importantly, those genes you think in your system is important. Then you will have a quick idea about this cluster, what they are, the cells supposed to be, and so you start to have an idea about your cell type. So that's the way to look at the genes. Okay. So this gene almost not expressed. There's only this cluster have some expression, and this small cluster. So this is a lowly expressed gene. All the other cells have zero expression when the color is gray. Okay, any other questions? You can have some other statistics about the cells. For example, we can have label size color coded. Something wrong, so forgot about this. Maybe. Yeah, so there's a problem to use this button, but I will try to fix next time. There are others, so you have a selection of those color maps. Make font size bigger, smaller. Okay, they all available. And to save the pictures, so you can save this whole thing into a JPEG format. Okay, the dark mode. I think that's all for this week. So next week our topic will be let's see, clustering. So once you see this and you know there's clusters of cells, what technique we can use to help you to group them into different groups. Okay, that's for next week. I leave the rest of time for your questions and answer any individual questions. I will work around and see. Okay. Everything is fine.